So thank you, Gothenburg and West Sweden, for that inspiring, magnetic explanation of your fantastic, or our fantastic region. Now we are turning to the next item on our agenda. We're going to listen to some fascinating things about present research with the title, We Know What You Eat. It sounds to me like Snowden or something. Somebody who's watching our private lives. But this is about cutting-edge technology for measuring nutrition. And to educate us on this is Professor Ann-Sofie Sandberg, head of the Food and Nutrition Science Division at the famous Chalmers University of Technology in Gothenburg. She's an extremely productive researcher. She has co-authored 150 research papers, and of course only measured by international peer-reviewed journals. I tried to learn what it is all about, but I gave up because they were too difficult to pronounce the different subtopics. So we must listen carefully now. And I would also like to mention to you that she is a member of the Royal Swedish Academy of Sciences, which means that all of you who aspire for a Nobel Prize must be particularly kind, because that's the kind of things they do at the Royal Academy of Sciences. Now, welcome, Asi, as you were called when we were both young.
So, thank you very much, Leif Pagrotsky, whom I actually have known since many years. And I'm very happy to be in New York, and I would like to first thank the organizers for inviting me. And now we are going into research, and I will give some examples of the research we are doing at Chalmers University of Technology. So, why is it so difficult to measure nutrition? Because we want to know exactly what you eat, and traditional methods often does not give us enough information, or not the correct information. And then there is also thousands of compounds in foods which may interact with each other or with the body. And the effect of diet is mediated via multiple biological pathways in the body. And then, depending on genetic differences, individuals respond different to foods. Already Galileo Galilei said in the 16th century, measure what you can and make measurable what is not so. And that is what we are trying to do. So what do we exactly want to know as a researcher? We want to know everything in detail. When you eat your food, how much, what kind of food exactly. And as you can see, it's quite difficult. And how are that normally measured today? Yeah, it's to ask people, and using 24-hour recalls or dietary histories or diaries. So how many of you could, for example, remember exactly what you ate yesterday or 3 weeks ago or half a year ago? You see that it's not always the case that you get the exact answer, and then also people know that what they actually should eat, and they maybe don't want to tell some things that they should not eat. So what we need is some objective measurements of food intake. So what if we could measure what people eat without asking them? To take a blood sample and look into the compounds which are there, to use what is called dietary biomarkers for food intake. So that is what we are doing. And something called metabolomics, when you measure a lot of small compounds in the blood, several hundred compounds, then you can look for new biomarkers for food. For example, differing between a sourdough rye bread and another bread, or finding that you have drinking coffee or taken citrus fruits or fish by particular molecules. And I will just show you some examples what we are doing at Chalmers, and this is by one of the persons working with us, Alistair Ross, and he is working a lot with whole grain, and there we have found very quite validated biomarkers for intake. So we can, by analyzing blood samples, see if you have eaten rye or if you have eaten wheat, whole grain, for example, and by just measuring blood samples. What about meat then? Meat has maybe some associations with disease, and we want to know if people have eaten meat. But meat biomarkers are a challenge because meat is so much like ourselves in composition. So we did a trial where we measured after a meal, comparing herring and pickle herring and meat with matched diets, same amount of macronutrients, only the protein source then differed, and measuring with metabolomics around several hundreds of compounds in the samples, and taking blood samples from these subjects after different time points. And what we could find then was actually what we think will be a new biomarker for meat intake, some amino acid called beta-alanine, which was much higher than after intake of beef, minced beef, compared with baked and pickled herring, and also for hydroxyproline. So we think that is a potential biomarker for meat intake. But we also want to know and to measure what the food we eat does to your body, to our bodies. And then metabolomics is also a very good tool, because it tells us what has happened as a result on effects from genetic level and protein level, and then we can look at the metabolomics in the blood or in the urine. And now you will see a picture which is very difficult to read, and you don't need to read it. I just wanted to say that we can, at the same time, we have developed a method that we at the same time can do both untargeted and targeted analysis of a blood samples. We can scan for several hundreds of compounds in the blood, and at the same time measure and quantify specific compounds which we want to look at, so that we can get much more information than before out of a blood sample. So one question that we can ask ourselves: should we eat fish or meat? And as you might know, high meat intake is often associated with increased risk of cardiovascular disease and colon cancer. And fish is recommended as a protein source. And you have surely heard about the omega-3 fatty acids, which are considered to protect against cardiovascular disease. But we are looking for other things also in the fish, because a fish, this is a herring, is much more than omega-3. Actually, 99% around about in a fish is something else. So we wanted to see if there are new effects related to the fish intake. So what we did a study, an intervention study with what is considered as the best way of doing intervention studies, that is to use crossover design. So the first 4 weeks, the subjects took a herring diet and they had a washout period, and the next 4 weeks, a chicken and pork diet. And it was only their lunch meals, it was 5 meals per week, and they were matched except for the protein source. And the other half of the subjects were starting with a reference diet and then ending up with a herring diet. And of course, we looked at all the blood lipid profile, inflammation markers, and we measured metabolomics. And now we come to something new and something very complicated. And I'm not going into detail in this, but what we found by doing metabolomics and doing what is called RIM analysis, relative importance of metabolites, in collaboration with systems biologists, we could find that there was a change in central energy metabolism between the chicken pork diet compared to the fish diet. And also we found a change in the amino acid metabolism. And what we then think that is connected to why people going on a fish diet actually lose weight, even if they have the same calorie intake. So fish is good. And another thing we found was also in the male subjects here, that there was an increased nitrogen oxide. And that is something positive because after fish intake, because nitrogen oxide is then reducing the risk of cardiovascular disease because it's vasodilatating. We used the same study as I mentioned before to look at the post-meal effects of eating fish versus meat, also to look for biomarkers for disease. And it was matched diet, same amounts of macronutrients, and it was only the protein source which differed. And what we found here was something related to diabetes type 2. Namely, we found after the meat intake compared to the fish intake an increase in 2-amino-adipic acid, which is considered to be an early biomarker for diabetes type 2. So this is something we are going to dig more into, but we found it very interesting. What we also are starting to do and want to measure is what is called personalized nutrition. Today, current dietary advice is, so to say, one advice fits all. It's done for an average person. But as I mentioned before, different individuals respond different to foods depending on genotype and metabolic phenotype. So we want to be able to predict what different people should eat, to advise, maybe not in the first step on the personal level, but give advice to groups of similar metabolic phenotypes. So one of the professors at our division, Rika Landberg, has started a project to look for different metabolic phenotypes, so-called metabotypes. And what we want to know then is, can we define these different metabotypes and find biomarkers for these metabotypes, and then tailor dietary advice for these different groups of people. And that is, of course, a challenge. And what also is a challenge is to give the biological explanation for these different metabolic phenotype. And the background there is both the diet, the genotype, the microflora. So we have to work with computational biologists or computational nutritionists to explain how this works. So this is what we hope to be able to do in the future, and we think that the novel tools in food research can give the answer what is healthy and why. And I want to show a picture of the people I work together with. And you can see also in the right corner there the two guys, Alistair Ross and Otto Savolainen, who has been responsible for building up this metabolomic infrastructure. And you can also find Richard Landberg to the right there, and he is a new professor working with personalized nutrition. And I would also like to mention that we are part of the maritime cluster of the West Gothia region, because we do a lot of research which is related to seafood and health effects. Thank you very much.